I suspect that not all of you are scientists. Nevertheless, I hope we can tell the story tonight in a way that you can all go home appreciating the principal message of our talk, which is that unlocking the secret of how cells die has led to this amazing new cancer drug, which is saving many lives, and I think has a very, very strong future in cancer treatment. And I want you to know that Melbourne scientists and clinicians have played a very, very important role in the development of this new drug. But before we venture into that story, let me tell you a little bit about how our scientific journey began. Our undergraduate science degrees, as you've heard from Luan, were in different opposite sides of the earth. Mine was at the University of Melbourne, Jerry's was at Emory University in Atlanta, Georgia. But both of those degrees at that time led us to be absolutely fascinated by DNA. And here you can see the two wonder kids of DNA, Francis Crick and Jim Watson in 1953. They're standing there marveling at the amazing DNA double helix they had just discovered, that elegant molecule that's been called the Mona Lisa of modern science. And a new science had sprung up in the 60s to unlock the secrets of DNA. This was called molecular biology, and it aimed to understand how our DNA encodes our genes and how that information is passed into our bodies to dictate the entire life process. So independently, we both decided to train at the Mecca of molecular biology, the MRC laboratory in Cambridge, affectionately known as LMB, for Laboratory of Molecular Biology, still an absolutely peerless place. This image here shows the 6 founding fathers of the LMB, and by that time already 4 of them had won Nobel Prizes, and indeed Fred Sanger, who is here, was to go on and win a second Nobel Prize. Actually, at the LMB, 12 Nobel Prizes have now been won, so you can understand why it is that we wanted to train there. So I went there at the end of 1966 to do my PhD. And actually, when I look back on it, I'm amazed that this happened, because I wrote to Francis Crick and I said, can I come and do my PhD with you? I had no idea how audacious that was at the time, but it's totally amazing. He said yes. Although, to be honest, he wasn't my direct supervisor. I was in his department. And Jerry arrived 12 months later to postdoc with Fred Sanger. And he'd just finished his PhD with Jim Watson at Harvard. But little did Jerry know that I was really intrigued to meet him, because I'd given my very first scientific talk ever here in Melbourne on his very first scientific paper. The reason I chose that paper to talk about was that it was absolutely fantastic science, which took us totally new places. But then when I looked up the 2 authors, I found out they were still only PhD students. So I knew at that stage that even as a PhD student, you can transform scientific understanding. So I was lucky. Max Perutz, who's here and who was the head of the lab, he introduced us at his Christmas party because he knew we both loved skiing. And the rest, as they say, is history. Although I have to say that Jerry very nearly blew his chances. In the first conversation, he admired my beautiful British accent. British indeed. I was totally insulted. So, life together in science has been wonderful. We started working together in Geneva, and you can see that we had absorbed the hippie phase in London. We've still got some of those shirts. And we had a wonderful time doing great science in Geneva. We enjoyed great hiking and skiing and great food and wine, but eventually we had to settle down and set up our own lab. And we had to decide where we would do that. Would we go to the US or would we come to Australia? And I very much wanted to come back here, and Jerry decided to give it a go here, for which I'm profoundly grateful. And thank goodness we did give it a go here, because it's been great. Now, our experience at the LMB had taught us that it's only possible to do great science in a great environment. And we found this great environment by looking around, going to many places in Australia actually on our honeymoon, looking at labs to see whether or not they inspired us. And the place that inspired us most was the Walter and Eliza Hall Institute across the road. And that's what it looked like in 1971. Of course, it was very largely at that time devoted to immunology, following the path of the previous director, Macfarlane Burnett, Nobel laureate, and now his charismatic student, and now, then the current director, Gus Nossel. So immunology was what we were turning towards, because it was asking big questions, and because the work being done at this institute was world class. It was tough at first, I have to say. This was a complete change of field for us, and our progress at the beginning was glacially slow. In fact, we only got there by the skin of our teeth just before our first grant ran out. But the pace of discovery picked up tremendously, and everything became possible in the mid-70s because genetic engineering, or recombinant DNA technology, was invented at that stage, and we adopted that technology. So what did we do with that technology? Well, for the first 10 years we were studying how the immune system generates the billions of different antibodies that it uses to fight infections. How could this happen genetically? And the answer was astonishing. This is a complicated scientific slide, but all you need to understand is that, unlike most genes, antibody genes are encoded as multiple separate elements that have to be brought together to make a complete functional gene. And the way that lymphocytes, B lymphocytes, do that is by cutting and pasting their DNA. And these genetic elements can be combined in many different ways, and that increases the diversity. So this is basically the secret of antibody diversity. So I want you to remember this cutting and pasting of DNA, because that's a very important part of the whole of the rest of the story. So we had a great 10 years. It was not only great in science, it was great because we had our two daughters during that period, and that was a wonderful life experience too, still is. But although we had enjoyed very much our time in immunology, things were stirring in cancer research, and they would eventually change our direction totally. Oncogenes, or cancer-provoking genes, had been discovered in 1975 by Mike Bishop and Harold Varmus, and some of you may have been here to hear Harold Varmus speak at the Graham Clark oration a few years ago. Now, what these two discovered was that certain retroviruses that cause leukemia in birds and in rodents, the genes that are in those viruses that cause the leukemia are very closely related to genes that we carry in every cell in our body, and that all other vertebrates do also. Now, the purpose of these genes is not to cause cancer. They're usually very carefully controlled. They're very important in cell proliferation. But mutations can occur to them which render them into very dangerous cancer-provoking genes. So Jerry and I were totally fascinated by this new field. We read widely in the new field and tried to find a way in that would enable us to join this new vanguard. And we found that in the fact that many cancers carry abnormal chromosomes. And this was particularly true of the leukemias and lymphomas. And of course, we were studying I hadn't actually told you that. In order to study antibody genes, we'd been using lymphomas, B lymphomas. So we became aware that many of these lymphomas and leukemias carry very specific chromosome translocations. Now, what is a translocation? It's when two different chromosomes break, and instead of being sealed back together again to their real partner, they happen to be joined to the other broken chromosome, so creating two hybrid chromosomes. We knew that these translocations were very common in leukemias and lymphomas, but there was a very big argument raging. Were these a consequence of the cancer, or were they the cause of the cancer? And that's what we set out to discover. And we started to work in a particular kind of B lymphoma called Burkitt's lymphoma. And it has one of these chromosome translocations involving chromosome 8 and chromosome 14. And what really piqued our attention was that it was known at this exact place where this break point occurs on chromosome 14 is where some of the antibody genes are located. Of course, I've told you that antibody genes have to be rearranged in order for a lymphocyte to make antibodies. So we wondered whether or not that process could go awry, and that by some chance the broken antibody gene locus had been joined to an oncogene locus, and therefore that had caused the cancer. So what we did was to take the antibody gene as a probe, and we isolated the DNA across the junction and searched for this putative oncogene. So really this was a scientific dream, and sometimes dreams come true, because when we did clone that junction, what did we find there but a known oncogene? This was an oncogene called MYC. It doesn't matter why it's called MYC, but it was known to be in retroviruses, and it was known to promote cell growth and division. So what's happened in this translocation is that it has linked the MYC gene to very strong on signals that occur in the antibody gene locus. This is called EMU, this particular strong on signal. So what happens when cells carry this chromosome translocation is that MYC is always on and at high levels, and cells keep dividing even when they should stop. So we thought we'd solved the whole origin of Burkitt's lymphoma at this point, but we needed more than an argument by association. We needed to provide direct proof, and to do that we turned to another technology that was very new at the time called transgenic technology. And what this involves is injecting DNA into, using a very fine pipette into a fertilized egg, mouse egg. And sometimes with low frequency, that injected DNA incorporates itself into a chromosome in that fertilized egg. And then that egg can be implanted into a female mouse who's been prepared with hormones to think that she's pregnant, and the embryo can be brought to term, and it's carrying an extra gene, so it's called transgenic. So what happened when we created mice carrying that piece of DNA from the chromosome junction, the EMU-MYC DNA? Well, what happened was amazing. These are the mice. You can see they've sort of got this water wing appearance because they're very lumpy. All of their lymph nodes are swollen from a leukemia. Every single mouse before it's 12 months of age comes down with a B lymphoma. So this absolutely proved beyond a shadow of doubt that this chromosome translocation that activates the MYC gene is the cause of Burkitt's lymphoma. This is now a very famous mouse strain. It's used around the world for studying cancer development, and many other oncogenes have been identified using this particular methodology that we used for this story. So with that background, I'm now going to turn it over to Jerry, who's going to tell you the main story. So remember, DNA breaks, chromosome translocations, they're going to figure in the next part of the story as well. Over to you, Jerry.
So I'm going to talk about another oncogene, one that we've carried forward quite a long ways. The BCL2, a novel oncogene, was discovered, I think, in 1984 by 3 American groups by using the same technology that we had used with finding the MYC, translocated MYC gene. The gene that they found was just simply called BCL2 because it was the second one that they had worked on in B lymphomas. And it said nothing about its function. So like the MYC, by analogy with the MYC translocation that we had already analyzed, it seemed very likely that BCL2 would be an oncogene activated by the potent ON signal E-mu from the antibody locus. So it's very closely similar in principle to what we saw with the MYC translocation. However, the sequence of the new gene, BCL2, gave no clue to its function. Does it drive cell proliferation like other oncogenes? So that was the aim that we set for David Vo, then a PhD student, working in our lab. And that work led to the discovery that the BCL2 promotes cell survival rather than cell proliferation. The test was to put it into lymphoid cell lines that are absolutely dependent upon a particular growth factor called interleukin-3. So when you take away interleukin-3 normally from those cells, they die very rapidly. The second panel shows the situation if you remove IL-3, in contrast to the left-hand panel, which shows IL-3 supported cells. So the idea of the experiment was that we thought it might well be that BCL2 would promote survival of the cells, proliferation of the cells, sorry. And so initially the experiment seemed disappointing because it did not, in fact, support cell proliferation in the absence of IL-3. However, the cells were still alive. So remarkably, this showed us something much more important than we might even have expected or hoped, that BCL2 is a oncogene because it promotes cell survival, a very different type of oncogene from MYC. The next step in the story was carried out by a new postdoc, Andreas Strasser, who turned out to be a real powerhouse. And this involved trying to test whether BCL2 would actually be an oncogene in a tumorigenesis type of assay. So to do this, we made E-mu-driven BCL2 and then interbred that with a mouse that already had a MYC transgene. And then we monitored the mice, and the mice started to die with tumors. But the ones that only had BCL2, nothing happened in this time span, whereas MYC was a dramatic promoter of tumorigenesis, as Suzanne told you. But the combination of BCL2 and MYC was even more potent. Now, why might this be? From other work that has been done subsequently, it's known that MYC normally provokes cell death by a process called apoptosis when nutrients become limiting. But BCL2 prevents apoptosis and allows the lymphoma to keep growing. And that's why the tumorigenesis is accelerated. Furthermore, subsequent work has made it clear that many cancers carry mutations that inhibit apoptosis. In fact, inhibition of apoptosis is now regarded as one of the hallmarks of cancer. So what is apoptosis? It's a genetically programmed process for very rapid elimination of cells in a way that does not cause any inflammation. And the left panel here shows a normal cell with scanning EM and one in which it's undergoing apoptosis. And you can see that the parts of the cell have been sort of packaged into this membrane-encoded bodies, apoptotic bodies, which are then eaten by other cells in the body. So cell death, and that usually means apoptosis, plays vital roles in normal physiology. It's critical for sculpting embryonic tissues. For example, the web between your digits has to be largely eliminated so your fingers are formed, whereas some animals retain that web because they have a different lifestyle. It's important and critical, in fact, for the development and regulation of our immune system, and perhaps most important, it balances cell proliferation in all of our tissues throughout life. And that's particularly important in the tissues which turn over rapidly, such as the gut, the liver, and blood cells. As well as contributing to normal physiology, imbalance in cell death can cause different types of diseases. So a healthy tissue requires that, in fact, the birth and death of cells be kept in pretty close balance. As we've already seen, if you have more birth of cells than you do death, it can lead to cancer, or in fact also autoimmune disease in lymphocytes. In contrast, if you have much more death than you do birth of cells, you may well end up with a degenerative disease. Now back to BCL2. How does BCL2 influence this process? And of course, as you might imagine, one gene would not be able to do all the things that I referred to. And it turns out that one family of genes, the genes that are reasonably closely related to BCL or partially related to it, can do so. And this family comprises about 15 to 20 members in different species, and it actually can be considered as three subsets of proteins. The ones most similar to BCL2 we can refer to as the cell guardians. They include, for example, BCL2, a close relative called BCL-XL, and another one called MCL1, and a couple of others that I won't bother to mention at the moment. But perhaps most strangely, other family members instead promote cell death, promote apoptosis. And in fact, the critical executioners are BAX and BAC, and their structure is remarkably similar to that of BCL2. These little regions here called BH4, 3, 2, 1 are actually the regions where there's the most homology within the family. And you can see that the executioners, BAX and BAC, have got all the similar regions that you find in the pro-survival proteins, the cell guardians. So what about this third group, third subset of proteins? These are sort of distant cousins of BCL2. They in fact only have one of these homology regions called BH3, and we now know that that region is needed for protein association within the family, as I'll explain more in a minute. Two examples of these proteins are called BIM and BID. Notice, because it becomes quite relevant, that all these proteins have a region that are called here as TM, that's actually used for targeting to the mitochondria, because that's where most of the action occurs. I think I'll skip ahead a slide to explain the way in which the family works. The main thing that you have to understand about the interaction of these proteins is explained on this slide, which shows BCL-XL with a region of BIM that we call the BH3 domain, shown here as a yellow helix running right up this sort of groove in the molecule. So this is what the interaction, the key interaction in the family is. The BH3 domain of one of these BH3 only protein binds to the surface groove of BCL2 or its pro-survival relatives. And this neutralizes the capacity of BCL2 to inhibit activation of BAX and BAC pro-death proteins. Now I want to turn to the drugs that have come out of these fundamental basic studies that I've just been telling you about. They're called BH3 mimetics, and you'll already realize the reason for that, most likely. What they are is they're small chemicals, relatively small chemicals, that are able to act in a fashion very similar to the BH3 domain of BH3-only proteins. And I'll remind you so that cancer drugs, in fact, conventional cancer drugs that is, actually work by inducing these BH3-only proteins. So cancer drugs kill by activating BH3-only proteins to initiate apoptosis. But tumor cells often resist apoptosis by having defects in production of the pro-death BH3-only proteins or excess production of the pro-survival proteins. So nevertheless, if we could find a chemical that binds like a BH3 domain to the groove of a pro-survival protein, such as BCL2, this should prevent it from suppressing activation of the pro-death proteins back and back. So how did these so-called BH3 mimetics come into existence? The first triumph came from Abbott Laboratories, now called AbbVie, in 2005. In a Herculean 10-year feat of chemistry informed by structural biology, they were able to find and elaborate a really complex organic synthesis of a molecule that could fit into the groove in a way that sort of approximates to what the BH3 domain of a peptide does. As you can see, it's in roughly the same place. You can see here we see in purple the helical BH3 domain. In blue and yellow, you can see the parts of this drug which was first called ABT-737. And we immediately started using it to test it out. And we were able to confirm the results they had reported. It's a genuine BH3 mimetic, and like BAD, it binds strongly. That's a BH3-only protein called BAD. It binds strongly to BCL2, BCLXL, and BCLW, but not to two other pro-survival proteins. Of course, it had to be first tested in cell lines and in mouse patients, if you will, before it could go into the clinic. And we participated in some of those studies. But the real tests, of course, came when a closely related compound, also made by Abbott, called ABT-263, and the generic used in medical terms is Venetoclax, was a potent orally available compound closely related to ABT-737. And Andrew Roberts played a very big role in all the first studies with it. He's an expert on, among other things, chronic lymphocytic leukemia, one of the most common types of malignancies. And it was chosen for a study because it has high levels of BCL2. And in fact, the early clinical trials showed very encouraging results, suggesting that there were some good effects of it. For example, here's a striking result of treatment of a patient who's already failed 7 prior regimens, 7 different types of drugs tried on him before, and failed all of them, and has a large tumor, which after 7 cycles of treatment with ABT-263, it's essentially almost disappeared. This is not a cure, some residual disease detectable, but it seemed to remain stable. Unfortunately, that drug turned out to have a dose-limiting toxicity. And by that, I refer to the fact that platelets, which we need for blood clotting, it turns out depend on BCLXL for their survival. And Venetoclax binds to BCLXL as well as BCL2, and therefore can kill platelets. So it's what we call an on-target toxicity. The drug is working like it should work, but unfortunately that limits what can be done in the clinic. This limits the tolerable drug dose that can be used. So this was one of the incentives that led us, our institute, WEI, to enter a collaboration for 4 years with 2 of the big guys, Genentech and Abbott, in 2007 to 2010. And this collaboration produced the first BCL2 specific target, called initially ABT-199, and now known by the generic name Venetoclax. Some of the early results were absolutely astounding. This is the first 3 people very limited start on a clinical trial, but this is the 3 first people treated with a single oral dose and over 95% drop in the number of lymphocytes within 24 hours following a single dose of ABT-199, including later a rapid reduction in palpable lymph nodes. There was even some evidence of rapid tumor lysis, which can actually be extremely dangerous. And so subsequent experiments, clinical trials, were always carried out with a more gradual build-up of the level of the drug, because it essentially was working too well, causing so much lysis of the tumor cells that, in fact, leading to one death. And as expected from what I've said before, it only had a minor impact on platelet levels because it is absolutely specific for BCL2. And you see the impressive shrinkage of lymph nodes that occurred after weeks of therapy. This patient is still in complete remission with treatment started in 2013. Of course, these anecdotal things prove very little. What you really have to do is have serious clinical trials, and that's exactly what Andrew Roberts and colleagues did. They carried out clinical trials of Venetoclax alone for chronic lymphocytic leukemia. And this actually, their trials, the success of those trials led to its approval by the US FDA. After some months, most CLL cells were eliminated not only from the blood, but also from the lymph nodes and the bone marrow. Terrific results where you can see that in multiple patients, you're getting a great drop in the tumor cells, almost complete, albeit not always, not usually totally complete. The result was that in April 2016, these successful trials made the FDA approve Venetoclax for treating refractory CLL in patients with a deletion on chromosome, the short arm of chromosome 17, which is usually involved in loss of the p53 tumor suppressor. So I just have to address one more question, but a tricky one. Why are cancers more susceptible than normal cells? We don't know the full answer to that as yet, but here's a plausible model for why it ends up working. We think it's mainly important to emphasize that the stresses in cancer cells are quite enormous, such as DNA damage, altered metabolism, hypoxia, and so forth. And this select for upregulation of BH3-only proteins like BIM. And these complexes build up and make the cancer cell nearer the apoptotic threshold. That is, they're primed for death. But Venetoclax can displace BIM from BCL2 because it actually works better even than this most potent BH3-only protein. So it actually displaces BIM from BCL2, allowing BIM to then attack other pro-survival proteins such as MCL1, and probably also to directly activate BAX. So when BAX and BAC are no longer restrained by pro-survival proteins, BAX and BAC can be activated, and they form both dimers and larger polymers that lead to apoptosis. Now Suzanne's going to finish off by talking about the progress on this drug and others since the FDA approval.
Okay, so we'll go fast, but there has been a huge amount of progress, and I think that you should understand that things are going extremely well. So we talk about monotherapy first, that's treatment just with Venetoclax, and we're talking about, again, treatment of patients who failed all normal regimens of treatment. It's a complicated slide, but I just want you to look at the column that's marked with red. And you can see here different B lymphomas, chronic lymphocytic leukemia, follicular lymphoma, and some others. And you can see it works well as a single agent to various extents. You get complete responses in a trial of about 20% of patients with chronic lymphocytic leukemia. It's also very good in mantle cell lymphoma as a single agent, complete responses in about 20% of cases again. It's also showing promise in follicular lymphoma, although you need higher doses of the drug for follicular lymphoma, and it's got some activity in another class of tumour called diffuse large cell B lymphoma. So it was working pretty well, but not perfectly as a single agent. The next logical step, of course, was to try combination therapy with other drugs. And I'll just quickly whiz through a couple of these that have now gone through substantial clinical trials. One of the combinations trialed was Venetoclax plus rituximab, which is a monoclonal antibody that had already been approved in the clinic for treating B lymphomas. And here you can see you're now getting complete responses in 41% of the cases instead of just 20% when it was used as Venetoclax alone. I should emphasize here that you need prolonged treatment with Venetoclax to get these very good results. But here's another very interesting set of data from these first trials. 11 patients went off Venetoclax after they'd been treated with it for some time. And 11 out of the 11 remained totally free of residual disease. So that gives us hope, I think, that at least for certain patients, combination therapy with Venetoclax may eventually lead to total cures, which would be totally remarkable. Another thing we haven't told you about these drugs is they're very, when you get treated with normal chemotherapy, all sorts of nasty side effects occur. These drugs are very well tolerated with very few side effects. So 11 out of 11 patients not progressing and not having gone through all of that trauma from normal chemotherapy is a very, very fine result. And then there's another drug that had already been approved and Venetoclax has been combined with. It's called ibrutinib. It doesn't really matter what it does. What I want to tell you is that now they're getting 63% complete responses in mantle cell lymphoma, whereas I told you before it was about 20% with just Venetoclax alone. And it's up to 51% with this combination for chronic lymphocytic leukemia. So now there's a trial being planned with all three of these drugs, Venetoclax, ibrutinib, and another anti-CD20 monoclonal therapy, and we look with great hope towards the results of that. Venetoclax is also looking very promising for acute myeloid leukemia, which is a very, very, very nasty disease of which there's been practically no improvement in treatment for decades. Elderly patients can't have bone marrow transplants, but they have responded very well to a combination of Venetoclax plus azacitidine. And again, 60% complete responses and a median overall survival of more than 18 months. Really promising results. And as a consequence, the FDA has approved this combination for treating AML patients in the elderly. So Venetoclax is now in more than 50 clinical trials worldwide for blood cell and other kinds of cancers. And remember, Venetoclax, as Jerry told you, is BCL2 specific. What about the other BCL2 family members? Can drugs be developed against them, and would they be helpful in cancer therapy? Well, the one standout target is MCL1. Why is it a standout candidate? It's because lots of human tumours have got amplification of this gene. That means many more copies of it. And in particular, relapsing tumours often show very high levels of MCL1 protein. And we know from experiments in the lab that high levels of MCL1 confer resistance to Venetoclax. And it was a very good bet that drugs that are targeted against MCL1 will be hopefully very helpful in the clinic. And so that's the reason why we high scientists have been collaborating with a French company, Servier, to develop a very promising MCL1 inhibitor. It was a much harder task even than developing the very first BCL2 inhibitors, but there are now at least 3 different companies with very promising inhibitors of MCL1 in the clinic, and we look forward to seeing the results of that. Okay, so we've taken you on a 30-year odyssey. And why does it take 30 years to get where we are now? There has to be a discovery phase, and that's the discovery of the gene and how it works. And that's an ongoing discovery phase, still going on. We still need to understand more about this BCL2 regulated cell life-death switch. Then there was the translation phase when WeHi initiated its own BH3 mimetic program, and Abbott had its own program and published the first bona fide BH3 mimetic. In 2006, we haven't told you, but because we had our own BH3 mimetic, we could broker this deal with Genentech and enter into this collaboration. And we eventually had this unique 3-way collaboration. And then, of course, there's the very, very, very important clinical phase, which takes many years to accomplish. So that's why it took 30 years. I wanted to finish by saying WeHi was extraordinarily lucky to have been part of this story, and what have been the key lessons that we've learned from this. I think the key ingredients in our success in this program were first scientific expertise. We had collaborating lab heads with great expertise in the molecular and cell biology of cell death and also of cancer. They were joined by other really internationally magnificent scientists in structural biology, medicinal chemistry, and drug development. Without all of these scientists working together in different disciplines, we would not have been competitive in this very competitive scene. Other key ingredients at WeHi were we had a really excellent business development office headed by Julian Clark. He had tremendous experience. He brokered a very, very difficult negotiation with Genentech to get eventually WeHi having a small percentage of the sales from Venetoclax, which we're ploughing back into further research. We also had invested in a high throughput chemical screening facility and medicinal chemistry labs, without which were headed by Ian Street, and without this we could not have participated. And of course, we had tremendous clinical trial expertise headed by Andrew Roberts. And I wanted to emphasise here how important the Victorian Comprehensive Cancer Centre has been, because it has tied together fabulous clinicians from the Royal Melbourne Hospital and Peter Mac in this endeavour. And finally, really importantly, what has been very important was that we had long-term funding for basic science. We had that from the NH and MRC, we had it from the US, from their Leukemia and Lymphoma Society. Atlantic Philanthropies helped us build these labs that were so important. And of course we had very important funding from both the Victorian and the Commonwealth government for infrastructure. So I want to finish with this message that the success depended on strong, long-term investment in discovery science. You know, there's an awful lot of emphasis these days on translational science, but without that really long period of discovery science, there would have been no pipeline, there would have been no cancer drugs. So without discovery science, there's no pipeline either to business or into the clinic. And I want you to tell that to every politician you meet. We scientists know it, they don't know it. So thank you for your attention. Thank you.